Hello and welcome to Nikolai's Genetics Lessons, and in this video I'm not going to give you any problems to solve. Instead I am going to explain what is a retrogene. And I choose this topic today because I didn't find any information about retrogene on the YouTube, so this is going to be the first video on the YouTube about what is a retrogene. In this video I'm also going to explain importance of retrogenes, and in the end I will talk about perspectives of the genetics in the near future. So I would start from afar explaining the central dogma of biology. And central dogma of biology that we know about now more than 60 years tell us that gene expression follows uh, like this. So first we start with double-stranded DNA, and then message RNA would be made, single-stranded. So we start with DNA, double-stranded, and then single-stranded message RNA, and then this message RNA would be used in order to be uh, translated into the protein, and protein would take then three-dimensional form and may also aggregate with different proteins to become fully functional. And this called central dogma of biology, how genetic information flows and how proteins are produced. And we as humans and any other animals represent protein form of life. So this pathway here is very important and called central dogma of biology. But about 10 years later, after Francis and Crick discovered the structure and function of DNA, other scientists have found that different pathway also exist. Some cells can be infected by virus, RNA virus, and RNA would be used in order to produce double-stranded DNA, and that double-stranded DNA would be inserted into the uh, double-stranded DNA of the host organism. So we call such viruses. I will use different color so you would better see. And such viruses we call retroviruses. In order to make double-stranded DNA from the single-stranded RNA, such viruses also carry such proteins that we call reverse transcriptase. And this enzyme would make a second strand, complementary strand of the DNA. So on the first stage we would have a hybrid between RNA and DNA. Then RNA would be degraded and double-stranded DNA would be made, and one of the DNA strands would be used as template in order to make the second strand of the DNA. And then, as you see, it would be inserted into the genome of the host organism, and later such viruses can express itself under influence of different conditions, for example, stress or any other environmental triggers. Once again, we call such viruses retroviruses. And now you're ready to understand what the retrogenes are. Imagine that any given cell would express thousands of genes simultaneously, and if a virus would infect such a cell along with a genome of this cell, some of the genes would be expressed, and also we can see here genome of the virus, but also, as you remember, virus would carry such proteins that we call reverse transcriptase. This is enzymes that allowed reverse transcription of the RNA into the DNA. And in some rare cases, one of such enzyme can make a double-stranded DNA using the host RNA. As you remember, after message RNA would be expressed, it would be in its immature form, so would consist of both exons and introns. And later, introns would be cut away, and we're going to get mature form of the message RNA. So only these fragments would be present in the mature form of the message RNA. So if this fragment exon 1, this is exon 2, and this is exon 3, in mature message RNA we would find all these three fragments present, but introns would be absent. Now imagine that such mature message RNA were used by reverse transcriptase of the virus, and second DNA complementary DNA strand were built, then message RNA were degraded, and second complementary DNA strand were built. So now we have double-stranded DNA, and by repair mechanism of the host organism, such fragment can be, or such gene can be inserted in any place on its own genome. So now I would use these lines to designate new place of this gene. We can find such gene in any place of the genome on the any chromosome. So now we call this retrogene. Retrogene. And we can designate it from original gene because it would have the same sequence but without introns, and we would call such gene retrogene. And of course another variant of the same gene have to be present, that variant that includes introns. Now about importance of retrogenes. Nowadays scientists have sequenced many genomes of the many mammals, and they have found that mammals has about 20% of the genes that represent retrogenes, or genes that doesn't have introns. And this is very important for evolution. So evolution goes not only by mutation of the single nucleotide, but also by duplicating whole genes. And we have many such duplicated genes in the form of the retrogene, and also some of the genetic disorders and disease we can connect to retrogenes. And here is one of such examples. By breeding and selection, people got some breeds of the dogs that we can call dwarf dogs. So this is normal sized dogs and this is dwarf dogs. And now when we can compare genomes using wide genome association method, and scientists have found that one of the genes in dwarf breeds of the dogs are present and absent in normal dogs, and this is retrogene, and this retrogene is called FGF4, and actually it regulates another gene that is called FGFR3, and the gene leads to very short limbs. It is very early calcinated those limbs and bones in its development and don't develop full size, and the same gene FGFR3 in humans also leads to dwarfism. But the difference is that in humans, uh, mutation in regulatory sequence of FGFR3 gene cause this up expression of this gene, but in dogs we have extra copy as retrogene that regulates expression of FGFR3 gene, but as you see, the result is the same in humans and in dogs. It leads to this form of dwarfism. And now the question is whether this is a recent event, this mutation in the form of retrogene inserted in the genome, or if it were long time present in dogs. Actually, it is long time present in dogs because this form of dwarfism is very rare, but also may happen in wild animals. But of course, wild animals would die and wouldn't be fit in the environment, but artificial selection of dogs for whom humans would care lead to fixation of this retrogene in certain breeds of dogs. And now I will talk about the perspective of the genetics. If you remember, maybe about 20-30 years ago, programists, in order to, for example, make a ball move from the left side of the screen to the right side of the screen, you have to be programmist. But nowadays, of course, any school child can do many manipulations on their screen using special programs without extensive knowledge of the how programs are work. So nowadays we are in genetics on the same stage. In order, for example, to make one breed very short-legged, we know what have to be done. We have, for example, to introduce FGF4 gene, but you have to be genetist in order to do this. But imagine that in the future, everything you have to do is just create a picture on your computer, and for example, you want the same breed but short-legged. Everything you have to do is just to manipulate the picture, and the software would instantly know that you have just to introduce retrogene in order to achieve these results. And you can manipulate color, you can manipulate fur, and even intelligence. As you know, for example, Pekingese are very stupid dogs, but some of the breeds are very really smart dogs. So imagine that we would be able not only use such phenotypic characteristics as appearance, but also such as intelligence. So this would be achieved by, I think, in 20-30 years, that even those people who don't anything about genetics would be able to create new breeds just using their computers, and this would be as easy uh, as nowadays children can manipulate pictures on their computers using special drawing softwares. And when you would be satisfied with the picture and description of behavior, you can send a sequence to biotechnological company that would introduce these mutations, or just would combine existing known fragments of the dog genome that would lead to such changes and would create a new breed for you without extensive breeding, program that may take many years. It can be created within just months. The other good side of it would be that, as you know, many breeds nowadays has many genetic problems, health problems, due to close relative breeding, when mother would mate with her son and father would mate with her daughter and brother and sister would mate. This is how selection works in order to fix some characteristics, but also this would fix some genetic disorders in its homozygous recessive form. So as you know, most of the genetic disorders, I would say about 99% of them, are homozygous. So you need two genes in homozygous form in order for the genetic disorder to be phenotypically expressed. So most of these breeds has also health issues. And now we can just make those sites on the genome to be whether homozygous dominant or at least heterozygous, and get rid of all these health issues with these dogs. You may think that only these breeds should have specific health problems, but such breeds as this one shouldn't have, because it looks almost wild wolf. But actually, as you see even on this picture, these breeds suffer very frequently from hip dysplasia, and actually we would be able to eliminate all these health problems in just 20-30 years from now, and even children would be able to create their own pets for their birthdays. Just think about this. This is absolutely mind-blowing idea how our life can be changed in just 20-30 years from now. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any, and see you in the next video. Goodbye.